Hi, I'm Tom Ouldridge, and I'm interested in how molecules perform complex tasks like self-assembly, locomotion, and information processing. Tasks like these underlie all aspects of life, including DNA duplication and cell division. My group is highly interdisciplinary, stretching from computer scientists to experimental biologists via theoretical physicists. We use theory to probe the fundamental principles of these systems, identifying how the laws of physics determine the minimal ingredients required for a task. We then test our understanding of these principles by designing and building our own synthetic versions of the vastly more complex natural systems. The process invariably deepens our understanding of the basic physics and throws up new theoretical questions to investigate. We're constantly amazed by what natural systems can achieve, for example, in their ability to perform complex computations at low energy cost. Studying them the way we do has three benefits. First, we test and expand our understanding of biology, just like the best way to understand how a bike works is to tinker with one or even build one for yourself. Secondly, our work makes fundamental questions at the heart of physics tangible. It's much easier to think about the physical limits on information processing if you have an actual device in mind. Thirdly, and just as importantly, the output of what we do are functional molecular systems that can be either plugged into existing biology or used to engineer lifelike systems from scratch. Imagine what we could do in industry and medicine if we could couple the power of life with the control we have in, for example, electrical engineering. I'll let one of my PhD students, Javier Cabello Garcia, explain what he's doing.
Did you know that inside every cell of our body there is a small vault? We call it nucleus, and like every vault, it contains something very valuable, all the information necessary to create a cell, and by extension, us. This information is stored in double helices of DNA, encoded as a sequence of characters. Each human cell contains 6 billions of these characters in its DNA, which can encode 2 gigabytes of information. That may not look impressive, but if you consider the approximately 30 trillion cells in your body, that means that you are walking around with 4 times the data in the whole internet, just encoded in your DNA. But believe me, this information is more relevant to you than the internet, because your DNA sequence is what makes you look different from your cat. So the main reason why you are the same species as your parents is because cells perform the titanic task of copying all the information into your DNA, using your parents' DNA as a template. But the copying doesn't end there. The information in DNA is constantly copied to create proteins, the small biological machines that the cell uses as tools to carry out its functions. The aim of my PhD is to understand how copying processes work and their origins. This is not an easy task, because for copying, the cell requires many different proteins that interact in ways that are not very well known. In addition, the origin of copying is unknown as well, because if you need proteins to copy the DNA sequence into more proteins, how was the first protein made? Answering this chicken and the egg dilemma will reveal how DNA sequence was copied before cells and proteins even existed, which is the same as discovering the origins of life as we know it. Given the dimensions of this task, during my PhD, I did as the philosopher Descartes says: to solve a complex problem, you must reduce it to its minimal parts. So my work consists in removing all the protein from copying processes, and prove if it is possible to build an artificial DNA copying system only with a minimal amount of pieces, just a template with a sequence and the pieces that will form the copy. At first glance, this can look easy. Just by mixing template and DNA pieces together, the DNA pieces will bind, just like a magnet, to the complementary sequence in the template. Then, the pieces join, producing a copy with the correct sequence. But there is a catch. The produced copy is so strongly attached that the copy cannot detach on its own. It's not autonomous. So to finish the copying cycle, the classic approach is to increase the temperature so the copy can detach, and then decrease it to produce a new copy. Imagine a cell working like this. It will be like a wind-up toy that needs constant intervention to keep working. During my PhD, I discovered a new mechanism. In my design, DNA pieces bind sequentially, and the binding between pieces produce the detachment of the previous one, just with a lever mechanism. This way, the binding between copy and template is weaker, so copies can detach autonomously and keep being produced as long as there are pieces of DNA remaining, just like a real cell. This way, I built a minimal system capable of producing copies autonomously, bringing us closer to understand copying and determine what was first, the chicken or the egg. And who knows, maybe create artificial life. Many thanks for listening.